bio. Dr. Brown earned her Bachelor of Science with honors in the Ecology Department at the University of British Columbia. And then she followed that up with a PhD in molecular evolution in the Department of Zoology, also at the University of British Columbia. And then she did a postdoctoral research assistantship position in population genomics at the University of Montana, in the Division of Biological Sciences. And then followed that up with a postdoctoral fellow in comparative genomics at Oregon State University with Dr. D. Denver. She then took a position, her current position at Texas Tech University in 2016, I think, is that correct? And Dr. Brown, her lab explores how innovation emerges from the microbial symbiosis using high-throughput genomic and transcriptomic sequencing, bioinformatics, population and comparative genomics, confocal microscopy, phylogenomics, and microevolutionary analysis. Her talk today is on the comparative genomic insights into the wonderful world of plant-feeding invertebrate microbiomes. Well, the title. Thank you, Dr. Brown. Welcome.
Thank you. So, thanks everyone for coming today. I probably will get to some of the highlights that don't make any sense in this slide later. We hope so. So, I just want to tell you about my driving kind of questions and reasons for being in this field. I'm really interested in the origin and the maintenance of cooperation. And so there's a long history and study of cooperation versus competition. We all know survival of the fittest, or the concepts that were brought to us through Darwin and others. And so we also know that cooperation is one of the forces in nature. But as a young person studying nature, it occurred to me that there's a lot of cooperation that's not associated with kin selection. So I used to like ants, and I'd say, why do ant colonies cooperate? Why do those little ants help? And they're related to each other. But there's a lot of cooperation or mutualism amongst organisms that are not kin, not closely genetically related. So this is really my burning question. Then over the years, and I'll explain a little bit about my past, I entered the plant world. And so today I'm only going to talk about a little bit of my research that focuses on the mutualisms that occur as a result of the narrowed niche in plant-feeding organisms. And what I'm going to argue today is that there's been convergent symbiont recruitment. So bringing in of new symbionts that do similar things in similar ways from independent origins. And that there have been convergent genomic changes. So independent. So one of the things about evolution that's interesting is seeing convergence, seeing independent cases of the same phenomena arising over and over again. And so as I said, I'm really interested in mutualism. And this is a little spectrum here from pathogens to mutualists, and some of the terms that we see are that they're opportunistic pathogens, organisms that are sort of neutral until in a particular situation, let's say a compromised host, and they become pathogens. There are also facultative mutualists. So these are organisms that may be, again, sort of neutral, but may help hosts under certain conditions. And so I'm interested in the drive, as you might say, or the trips that organisms go through this gradient in a short time span or in a macroevolutionary time. I'm also interested in who's controlling this movement through these different types of roles. And in particular, in my lab, we do a lot of DNA work, a lot of RNA work, and there's a world of microRNA. MicroRNA is an area where we can look at small signals that may control a crosstalk between these organisms. And so one of the phenomena that today I'm going to be talking about is genome reduction. And genome reduction is really commonly seen in host-associated microbes. And so we see here examples of, this circle represents a bacterial genome, which can be large, 4 million to 14 million base pairs, over time shrinking to very small size, and the smallest bacterial genomes are even in fact smaller than this. These are cellular genomes. So they're reduced far beyond the level that they would have to be for these to be free living. They only get this small when they don't have to do anything outside of the host cell. So these are intracellular reductions. So I'm interested in all this is happening. What happens to regulatory capacity? Are these microbes, as their genomes are getting very small, are they enslaved? And that's the current thinking, is that they're enslaved to their host. The host does all the control. There's not much regulatory capacity, and there isn't much adaptive capacity for change over evolutionary time. So those are kind of a lot of concepts to swallow right at the first of this talk, but I would like to kind of put that context out there. And we'll go through some more specific examples. So a little bit of the kind of pictures that come to mind for explaining where I'm coming from and why I study this. I've shown here some fish incrustation. This is actually what I did for my PhD work at University of British Columbia. I was looking at these little things, which are spore stages. And here's another one up here that live in hosts, in this case vertebrate and invertebrate hosts. And these are pathogens, and they're in the group Microsporidia. So they are a group of eukaryotic, highly reduced microbes. I don't study that anymore. I'm not going to talk more about it today, but it got me really interested in genome reduction because, as it happens, the Microsporidia have have the smallest genomes of any eukaryotic organism through long-term associations with their hosts. So there's still puzzles and mysteries as to how they do what they do and how they evolve. But we've moved, or I've moved now, into working on bacteria. And so today I'm going to be talking about bacteria that live in these hosts. Bacteria that live in 3 major groups of insects and also some in nematodes. And so I'm going to be focusing on the hemipteran insects. And these are cases where most of the time we have dual complementary symbioses. And I've just listed some names that won't mean anything to you yet, but this is an overview for later. And like parasitic nematodes. And there isn't a lot of work on their symbionts, but I'll hopefully illustrate to you today that we need more work on this kind of field. There's hints that there may be more to be discovered. And so this, it's a little bit dim from where I stand, but hopefully you can see, not to read the words on this giant phylogeny here. This is a review through Martin Kaltenkoff's group that just from last year. This has a really pretty picture of the order Hemiptera. So this is an order of insects, and this is the major clades in that order of insects. This includes cicadas, aphids, and others we're going to talk about today. But all the colorful lines are what I want to impress upon you. Each of these colored lines on top of the gray background is a group of bacteria. And the colors correspond to these major clades of bacteria. And what is maybe hard to see at a distance again, but if you can just maybe squint and trust me, on most of the branch tips, we see 2 or more symbionts. So here we see a red one and a blue one. You can't read the words, but it's a Salcia and a beta proteobacteria. And so this is very intriguing. And so there's been a lot of really recent work in this phenomena of complementary dual symbioses in the Hemiptera. And it all comes about because Hemiptera are piercing and sucking mouthparts. So they have a needle-like mouth apparatus with which they get into phloem or xylem, the juices of plants. And when they do that, they are feeding on a really nutrient-poor diet, particularly rich in carbohydrates, but very poor in amino acids. And so in this group, that's the focus. So how this evolves is another question, and how it metabolically, how we can look at it metabolically. And particularly, I find it really interesting that there's been replacement. So we see colors, but we see in lineages, this lineage all has the red lines. Hopefully you can see that. And most of the groups have a blue word beside it, or a blue line. But sometimes something else happens, where there's a loss of one of the bacteria and a replacement by another. And that replacement is really the crux of what I'm interested in. How does the host facilitate or allow other microbes to come and fill that gap? And to what extent is the microbe doing that? To what extent is the host controlling that? And so this is from another review by Angela Douglas. And this shows some more detail. So zooming in a little bit, this is a very unusual case. It's one of these complementary endosymbioses, but here it's a nested endosymbiosis. It's a bacteria inside a eukaryotic cell. And here we have 2 amino acid synthesis pathways, arginine and tryptophan. And on this side we have arginine, and what you see in the boxes are the enzymes required for that pathway, and what you see in the dots here are the metabolites, the substrates or the products. And so what you see is in the green is one cell, one bacterial cell, and the blue is another bacterial cell, and the brown is the host. And so what's happening is the metabolites are being shuffled across between these cell membranes, between these organisms, where the enzyme is present to do the next step. It's really crazy and unusual, but this is extensively found in this whole phylum, and this whole order, rather, of insects. So where I started working on this larger group was in a narrow set of these. These are Ishikawa is the name of a bacteria, and it's a symbiont who in 2007 a group in Japan, found actually drives the plant host preference of the host. So this is the host. It's Megacopter curvaria or Megacopter punctatissima, and these insects occur in different locations. This occurs in the mainland of Japan, and this occurs in the southern islands of Japan. Different phenotype, you can probably see different color there. They also have a different phenotype. This one, this is the graph that goes with it. This is its success or hatch rate on soybeans. This insect prefers soybeans, hatches better. This one here has a lower hatch rate, so it's considered a non-pest. In fact, it prefers a different legume. It prefers kudzu. Or you say kudzu, I think, instead, but I say kudzu. So, what's happened with this group is very interesting because these symbionts are not intracellular, so they're not endosymbionts. They're extracellular. And so what the insect does is it lays, these are its eggs, and it lays these little blobs, these little packets, which contain the symbionts. And these little sort of brownish things with the red dots, those are their eyes. These are the nymphs that are freshly hatched, and they have to poke their mouth part, their straw-like mouth part, into these little dirt-like blobs, which are packets of capsules. And they drink a sort of a bellyful of an inoculum of the microbes that they'll need to live. And what they're needed for is amino acid synthesis. These are phloem-feeding insects, and without those, the insects are very unhealthy, low fitness. So, because they're extracellular, you can actually remove these packets, sterilize the eggs, and swap them across these species. And what was done by this group in Japan was just that. And the result was that the phenotypes reversed. So these are this insect's results when they got the symbionts from this one. So their hatch rate on soybeans went down. So they became essentially, the way it's looked at, is non-pests soybean. They prefer kudzu. These ones had the reverse. These ones here, which are non-pests, when they get the symbionts from the pest, they become pests of soybean. They prefer. So the symbiont is driving the host preference. That's a nice little experiment. And that's where I began doing some research on the Kudzu bug, which was an invasive in 2009 in the US. So it arrived in Hall County, Georgia here, and has since spread to, I don't know the current count, but most of the southern states and all the way through Maryland, and I don't know how far north it is now. And it's following the distribution of its host plant, wild host, which is also an invasive from Asia, and that's the kudzu plant. So what the goal was with that project was to look at this. After the arrival of the insect, this is on kudzu, after about 9 months, it was found heavily infesting soybean and causing quite a bit of a loss, financial loss to soybean. And so the question was, is there a swap of the symbionts? Is a symbiont evolving or mutating in some way? Or do we have hybridization or what? And so we were trying to answer that hypothesis initially. So we did sort of, this is an overview of the way we did this work. We collected the insects on soybean and kudzu. They have slightly different phenotypes, which was very provocative. Slightly darker morphology on soybean in some cases, not all cases, and then the adults. They also had different patterns in their gut where they host the symbiont, different color. That was very interesting. So we also studied adults and nymphs. Nymphs won't have moved away from their plant yet, so they won't have fed on multiple hosts. And then we did a usual kind of pipeline. We isolated DNA and RNA, and then analyzed patterns. And this is the sampling scheme. So we sampled at ground zero, so we had archived specimens in 2009, and then we sampled for a few years thereafter. And we sampled DNA and RNA, and we found some novel mutations. So these are SNPs that we found. Ground zero SNPs were a little bit different, year 2 and year 4, without very much overlap. So we were seeing a lot of new mutations arise through this time. And one of the patterns that could be argued is something I won't get into in detail today, is allele surfing. It's a concept where there's relaxed selection at the wavefront, and there's increased chance, sort of drift at the wavefront of an invasion. Oops. So this is another thing that at first glance looks kind of confusing, but this is meant to represent the circular genome. So this circle here is the circle of the Ishikawa obligate symbiont in these insects. And these little bars going up and down refer to the allele frequency change from ground zero, from the first point in 2009 when the insect arrived. So we could do that because we had archived specimens. So here you see something where the allele frequency is mostly increasing all across the geographic range. In a few cases, that allele is decreasing. So that's the frequency going outward from the circle is increasing. And what we see going into the circle here, in a few places, there's stars, and we're more frame shifts. So frame shifts should be deleterious. They should go down. If the initial population has frame shifts that are, and genes that are important, well, those should decrease in frequency. So by looking at that pattern, we did find some enrichment. So we saw enrichment for several processes, including riboflavin synthesis genes. Riboflavin is vitamin B2 and is thought to differ probably quite significantly in different leguminous plants. And so this is kind of a working hypothesis that needs further work still, but that there may be this difference in riboflavin provisioning by the symbiont. Here's sort of an overview of COG categories. So these are just broad categories in function in the Ishikawa, and we have categories E and H, which are for amino acid and coenzyme, vitamin transport, and metabolism. And we saw a difference, so it's not shown with the star here, but E here and H were statistically significantly different depending on the plant. So we were seeing a difference, and then we also we're looking at non-synonymous change. So these are changes in alleles that make a difference in the protein, and that these were different based on plant, in particular for some of the amino acid and vitamin pathways. So in a very, very short time, in just 4 years, we were able to see differences in these allele frequencies. And then work in progress here is shown a bit here. With a collaborator at Emory, we are with Nicole Gerardo's group, we are working on the transcriptomics. So we have kind of an elaborate, nice, mostly nice design for sampling instars and adults on the two plants, sampling the whole insect versus the symbiont-containing part, which we call a crypt. And so we found preliminary data shows that the crypt and the body on kudzu and soy actually have differences. So there are differences in gene expression of the host. There are differences in gene expression of the symbiont. There's also Wolbachia in these, and every insect that we found has 2 strains of Wolbachia, which may be provisioning other things. We'll talk about Wolbachia in a couple minutes. So we did find overexpressed genes on soybean, including the riboflavin genes, suggesting, again, there might be some provisioning difference with that particular nutrient. So I'm going to then describe another group of hemipterans. These are the membracids. And I have to say, my work is early in this group, but they're really fascinating, both because they're kind of cute and beautiful. Here in these pictures, you see this thing called the pronotum. Does anybody know membracids? Has anybody looked at a membracid? We call them membracids. So there are not very many. They're neotropical. They're not very many in the temperate zone, but they're small. So you may be seeing them and not know you're seeing them. Anyway, this interesting structure here is kind of an adaptation that is an extension of the thorax here and wing and the forewings. So anyway, in Japan, Dr. Morimasa is one of the experts in. He's from Japan, but he lived in Brazil for I think most of his career, and he's one of the people who did extensive work in describing the diversity of these. And this morphology is, I like the title of this paper, The Art and Science of Describing Nature's Surrealists. And they're also surreal for their microbes. So I'll get into that. This is the first so far published genome of the microbiome, or in this case, a dual symbiosis in a membracid. And this is work that came out from Gordon Bennett's group in 2017, where here we have a plot of genome sizes, and we know that genome sizes get reduced against the GC content. And in different colors, we see different microbes. And in this color here, the smallest known bacterial symbionts, besides the very odd ones that were recently published in the Hodgkinia, which is a whole other story. These are cicada symbionts, this group. But what I wanted to point out, though, is that there's something wrong with this group. There are some branched chain amino acids that are not fully able to be synthesized from bioinformatic data anyway. And so there's a secondary symbiont, Arsinophonus, which may be jumping in and supplementing some of those missing enzymes for those amino acid synthesis pathways. But on the whole, almost every member of the order Hemiptera that feeds on phloem has to have dual symbioses And this has evolved over a long period of time. So what I was interested in with the membricids is that in 1943, there was an extensive study of membricids by a German group. And Agnes Rao published this work describing up to 6 different symbionts occurring in the symbiont organ, which I haven't described yet. It's called a bacteriome. We saw it in the previous figure. But this sort of different stipple drawing here represents those 6 different microbes in it. So this is quite an extensive and complex story here. And to my knowledge, no one's looked at these since 1943. So I applied for a little bit of money to study these, and got that money in collaboration with Monica Pupo and the University of Sao Paulo. And as soon as I arrived there in November to do this work, outside my hotel door were these are the nymphs of the membricids. And this thing feeding on them here is drinking the secreted sugar water, much like you would see in aphids that are tended. And this is a stingless bee, which are threatened in Brazil. And I had no idea that stingless bees did this. There are ants that tend these, but it was really impressive to see the stingless bee there, right outside my hotel. And so what we were looking for here was these target species that are in that list from 1943 that have 6 or more symbionts that nobody's looked at. So these are pictures that I found to help me running through the jungles, and these are what we collected. And so I was able to find, with great difficulty, a lot of those target species, and those are them. And up at the top are some of the ones you might see up here, including an invasive, the top there. That looks like it has a furry wool coat, and it's from Montana, where it's very close, from Siberia originally. So the work from the membricids is quite preliminary. I need to get back to Brazil and go and finish that work, but we'll do multi-omics and really characterize the metabolites exchanged. The next group in this same tree, where here we have membricids, membricids right here in the tree, is this funny dotted line here. This is the Typhlocybinae. It's a family or subfamily that has a dotted line on these figures because it's apparently lost all of its symbionts. That turns out to be not completely true. So the way that I got involved in this was when I first started in Texas, I went out to the cotton fields here and I did sweep netting and found these little green bugs and wondered, oh, they must have these Salcia nasui, the dual symbionts, let's just look at them. And it turns out we did a whole bunch of sequencing. We did RNA, microRNA, and DNA, and no symbionts. Well, I hadn't looked up the literature well enough and seen that they were on this dotted line. But we do see lots of little pieces of Salcia left over in the genome. I also put a picture here on Halloween last fall. I found these crazy faces. Can you see the scary faces? So I thought I would just share that with you for fun. These are another leafhopper that does have the symbionts. It has the 2 symbionts. But here it's like a lost soul trapped in the insect. This one looks like the screaming face with his hands up, doesn't it? Anyway, these made my day on Halloween to find these. And so here we are in the tree, the membricids, the Typhlocybinae near each other in the tree, very interesting. And so just to show you that the names are a little bit confusing here, the common names are leafhoppers throughout this section of the tree with the membricids in the middle. And so this is a sort of a little bit, again, a little bit faded color for today, but this is a pipeline of how we did our sequencing. So we actually dissect different sections of the insect, and then we do a whole lot with them. They're very tiny samples, but we aim to get some shotgun metagenomics, we aim to get some shotgun transcriptomics, and we do small RNA work. And then we do all this bioinformatics to put all the information together. And from that result, we have, here's this area, the bacteriome. These particular species, Empoasca fabae, which is it's called the potato leafhopper. We found it in cotton, of course. It has lots of different plants. And it turns out it has a bacteriome. It has an organ that would host these microbes without having microbes in it. So that's very fascinating to us. And we did some work. This is just showing you how much data we gathered. We did not find our Nasui or Salcia, these 2 bacteria, but we did find a whole lot of other microbes there that differed between head and thorax in some cases, and we found some pieces of Salcia. So here we found a cell wall-associated hydrolase from Salcia, a very good, strong match that was in this data set, but not allied with any actual bacterial cells. So a potential case of horizontal gene exchange. And so wrapping up all this insect work, I talked about the Ishikawa and the Kudzu bug, And I talked about the symbiote genotype affecting the host plant choice and also showing signatures of selection. So we saw differential selection depending on host plant or signatures of thereof. I talked about our early work in the membraces. So they have this accumulation of symbionts, and we're really kind of trying to trace down what's really happening. And that will be also performed with my Brazilian collaborator who's in pharmaceutical work, and we're hoping to grow a colony of one of the target membraces to actually be able to grow large numbers. In Typhlocypinae, they're a model for this loss. And we're really curious about that control that I talked about at the beginning. I didn't mention mosquitoes, but in my lab we're also interested in mosquitoes for a number of reasons. It's easier, probably, hopefully, knock on wood, to get grants to study things like Zika, which we're actually interested in. But mosquitoes are also nectar feeders. The males only feed on nectar. The females feed on nectar until they have that blood meal. And so one of the things that we notice in mosquitoes is that they're transferring microbiomes around. So in red here, I show you some of the questions we still have. How did Ishikawa confer this adaptive capacity? We really haven't answered that completely yet, but we're on to it. In the membraces, we want to understand the complexity. And in Brazil, where these ones have the greatest numbers of potentially cooperating microbes, we want to look at that and how it's associated with niche diversity. Some of these symbiotes may be defensive, maybe producing defensive compounds. And in the Typhlocypinae, we already see some potential horizontally transferred genes. So we want to chase that down. And so next I want to move to plant parasitic nematodes. And I forgot when I began to say thank you, of course, to my hosts, who are plant parasitic nematode researchers here. And it's the chronology of what I'm saying today is a little bit my chronology. I worked on the insects first, and then I bumped into, by chance, research on these plant parasitic nematodes, and I had very little prior experience, and I'm not a nematologist. So if I make mistakes about nematodes, you can correct me or take it with a grain of salt. However, the plant pests, I'm not sure why that's shaking. The plant pests we know are significant, of course. Plant parasitic nematodes are also significant, and together these can cost, I don't know if this is an accurate number. They also are responsible for I often talk to medical students in my lab at Texas Tech, and I try to get them on to study nematodes and insects, and they go, I want to be a doctor, why would I do that? I say, well, there are transferable skills in genomics, and also there's human health consequences of the way we control our microbes, and there's ecological consequences if we're using synthetic methods to control. So there's always an interest in less toxic methods to control our pests. But as I emphasized earlier, they're a convergent diet and feeding structure. So in the plant parasitic nematodes, they have this needle-like mouth part, which we call a stylet. Coincidentally, the people who study hemiparins have used the word stylet. It's probably not a coincidence. But these are convergent structures. They are not based on, as far as I know, not based on a history. They are similar morphologically in diameter, though. So both of these stylets are very similar in that they're both probably originated for probing into xylem and phloem vascular bundles of plants. And as a result of this probing into these juices of plants, the hypothesis is that these organisms were able to capture a new niche, have this new source of rich carbohydrates available, but at the same time having a little problem, which is the problem with the low amino acid content. And so there's parallel recruitment of microbial partners, and I'll show you some of those today. And so plant parasitic nematodes, some of you may have a better estimate. In case you didn't already know this, plant parasitic nematodes are very costly to crops. There are many described species that are polyphagous, feeding on lots of plants. And you may know this, that they secrete plant cell wall degrading enzymes, which are a very important focus, and effectors are a very important topic in the field. There are few microbiome surveys, very few and far between. And microbiomes may be important for potentially finding biological control. And I like to show this picture. It looks like a yogurt container. So you think about it as, oh, I'm going to take my morning yogurt of nematodes. Well, it's not for people. Beneficial nematodes, in this case, isn't plant parasitic nematodes, it's antipathogenic nematodes that you can use as a biological control for insects. So these pass a bacteria onto larvae of pest insects. And then I also show another fun picture, because I like to. This is the nematode art. Wouldn't you say that looks like nematodes? Does it look like nematodes? And where would it happen to be? It's on my walk to work every day. So my first day of work, I had no idea. This is a great big parking garage with nematode art all the way around, all sides of it. And I have no idea why it's there and why it's coincidental with me. So I just show it to you to be just as a source of amazement. Anyway, this is a kind of a schematic of a phylogeny for the plant parasitic nematodes with the plant parasitic groups in red wedges here, and in red names those genera. And what I produced here is those that have genomes available or limited, the black lines, those that have horizontally transferred genes or candidate genes very clearly within this group here that have a lot of representatives that are plant parasitic. Then we had here in red the sampling that I did when I was doing my postdoc in Dee Denver's lab. And those are these 6 species, and we picked them because of their importance in agriculture, and so many of them have really diverse host range. And what we did was this kind of a simple pipeline with the help of Inga Zasada and Dee Denver. We had various different collection strategies, and then we pulled together nematodes and obviously did sequencing. We also have a pipeline for skimming. So this is shallow sequencing, not deep sequencing, to kind of do sort of a first pass, very inexpensive scan to see whether there are microbes of interest. And this was some of the results. So these are our 6 species, each in a panel, and the colorful dots are the microbes that we found. And so this big splash, it looks like a volcano with an ocean here, and that's the lava, doesn't it? This one I'll talk about first. This is in Ziphinema. And this is another one we'll talk about, Protolincus penetrans, where we don't have a volcano, but we have these 2 clusters of colored dots right within the dots that are gray, which are the nematode dots. And those are separated by, in case you're curious, if you haven't seen these before, these are called blob plots usually. So we plot the GC content on the x-axis and the coverage, the amount of pieces that we get, and each dot is a contigue. So Ziphinema americanum is also known as the, I'm forgetting the common name all of a sudden, my brain's emptied that information. But the americanum is a complex of over 40 or so species that are very closely related. And it's in one of the earlier branching clades of the group. It's a vector of Nipoviruses, and it's important, and we found confirmation of this particular endosymbiont, Ziphinematobacter. It's a bit of a mouthful. We also recently see a Burkholderia-like symbiont in these. And so our questions were whether Ziphinematobacter is inducing parthenogenesis. That's being proposed. That's when males are not needed to make offspring in various methods, or whether there are other roles. And also, we want to see how widespread these are. So here's that volcano image again. There's Ziphinematobacter, and Pseudomonas fluorescens is this C, which I won't talk about at all today. And so we did fluorescence in situ hybridization microscopy, confocal microscopy. And in red here, the probe is to Ziphinematobacter. And this is the middle of the nematode, and it's the gut wall. And here we see the images superimposed, that's Staffy. And here we see the gut wall, when we move this around in 3D, is showing you that these dividing bacterial cells are rich and abundant in the gut wall. And they're also abundant in the ovaries, and also here's a nematode all curled up inside an egg. And here you see those developing bacteria. So the genomic side, in looking at an outgroup, this is an outgroup on the top, and here's Ziphinematobacter. The nearest outgroup that was available at the time has lots more genes, so these are proportional lengths saying the number of genes, and they're divided by COG categories, so it has a reduced genome and sort of generally proportional reduction in the different functional groups. This is a kind of a description, which I don't expect you to read all the words of, but it's the 2 outgroups. So this is a reduced Verruca microbia that's in soils. This is a reduced Verruca microbia that's in human gut, and this is our Ziphinematobacter. And what we see is enrichment for a number of nutrient provisioning pathways. And this is a lot of information too, but in a nutshell, the orangey colors are the essential amino acid pathways. These are pathways to synthesize, in black, the amino acids that animals, like nematodes and humans, cannot synthesize on our own. The other amino acids can be synthesized by humans, nematodes, and most things in between. So what we see in the dark colors is presence, and in the light colors is absence. So we see a lot of preferential retention of the essential amino acid pathways compared to the non-essential amino acid pathways. And this is a pattern we see again and again and again and again in the hemipteran endosymbionts of sap feeders. Here's a plot with similar data. So this is an enrichment plot compared to the outgroups, where the enrichment is represented above the line and the non-enrichment below the line. So we see essential amino acid synthesis genes are enriched in Ziphionomatobacter. And this is the distribution based on a literature review of the Ziphionema americanum group species. So all these are different species that cluster together all over the world. And these are all the ones that are now seen to have Ziphionomatobacter. So all over the world, this symbiont occurs in this group of nematodes. There's also a second symbiont, Burkholderia-like symbiont, which we won't talk about in great detail today. But I just wanted to mention that it's a potential case of what we saw in the hemiptera, a replacement of another symbiont. When we have an obligate symbiont that's lost, the host would drop in fitness immediately, and so if there's another symbiont present that's halfway there, it can sometimes pick up the slack, is the way we could phrase it. And so the conclusion so far from Ziphionomatobacter, does it do parthenogenesis induction? Probably not, because many Ziphionema species do parthenogenesis, though they don't have the symbionts. We've potentially shown, although it would be nice to confirm this with something, catch them red-handed, actually provisioning essential amino acids, but that's the hypothesis. So that this bacteria is provisioning essential amino acids, which is a little bit odd. Why would they be missing this unless it's a phloem feeder? Well, in the literature, we see very little data on Ziphionema americanum's feeding, but most likely it's feeding for some periods of time in or around the vascular bundle. So that's our kind of running hypothesis. We need more data on that. And we saw how widespread they are. And so we see parallel, I would argue, we see parallel recruitment of microbial partners between the hemiptera and these plant parasitic nematodes. So we'll jump to the last group. Cladolynchus penetrans is a nematode picture here, very distantly related to the last one. This one is in the Tylenchida. So these are major pests. We collected them, studied them from raspberry in Washington. And they're a really big problem up there because they require fumigation. They usually use methyl bromide, which is very problematic to use to control them in raspberry. There's some success growing these in culture, but that's still ongoing, so we can do more experiments. But as I showed you, we have something really interesting. These blob plots show two nested sets of colored dots. These are Wolbachia and Cardinium, two very distantly related bacteria that occur within these nematodes. And so we also see some others that I won't talk about today in the microbiomes of these nematodes. And so our questions. Well, for Wolbachia, there was one previous report here by Hagman et al. In 2009 of a Wolbachia and Rhodophilus similis, which is found in Uganda and it occurs elsewhere too. And nobody's been able to find Wolbachia in a plant parasitic nematode since that time. So it's been a kind of a contentious topic. So what do Wolbachia do? Wolbachia, for anyone who isn't familiar, they either form obligate mutualisms, as they do in filarial nematodes, or they're parasites. So they can be reproductive parasites. Strangely, the plant parasitic nematode Wolbachia was the first to evolve, as far as we can tell, relative to the outgroups. And the next are in these Pentagmonia. These are aphids. And so I would argue that this suggests that there was an early sort of plant dependency in the early part of the tree of Wolbachia. This is a tree, similar tree, with just a few genes that we could have used to incorporate more species. And here we see that Rhodophilus, the other one, from the clusters, as you might expect, nicely within the plant parasitic nematodes. The main message being that there are long branches, relatively long here. Wolbachia has been in plant parasitic nematodes for a long, long time. So I would argue that this says we're missing a lot of Wolbachia that may still be out there. There's a lot to be learned. So this is some details that I won't go into today for time, but these are some assembly statistics, some patterns with differences where Wolbachia is in yellow. But I think for time we'll just move on there. These are orthologs. So we see a lot of shared orthologs in the middle here, conserved between the reproductive parasites and the mutualists. we see our Wolbachia with a lot of unique gene groups that are not shared in the outgroup. And so we really focused on this group of genes to try to understand the function and the evolution of this group. So we don't have an answer to this question, really. In fact, it appears that our Wolbachia is somewhere between, at least from this data alone. This is a very similar idea. We have the proportion of genes shared with our Wolbachia with all of these other strains for which there's a full genome. And we see our Wolbachia clusters more. These are the mutualists, and they have a higher bar, which means that they're closer in gene repertoire, overall numbers of genes. And then what else is in there? Well, it turns out that if we compare in yellow our Wolbachia with the red and the blue again, the mutualists and the reproductive parasites, there's some similarities. Iron metabolism is conserved across the board, whereas our Wolbachia seems to be missing a lot of B vitamin synthesis. It seems to be missing the cytoplasmic incompatibility genes, and it's missing the WO phage, which has been an important focus for cytoplasmic incompatibility. And this is a lot of detail here, but this is that riboflavin gene. So we talked about earlier with one of the insects. Turns out our Wolbachia is missing most of the riboflavin synthesis genes, so that kind of dismisses the idea that riboflavin provisioning is an important part of this Wolbachia success. And this is work that's unpublished from collaborators where we look at whether Wolbachia might be distorting the sex ratio, which many Wolbachia strains can do. So here we see a correlation between the number of females, or the sex ratio, being distorted towards females, which is good for cytoplasmically transmitted endosymbiont, versus the prevalence. So the more Wolbachia you have, the more likely your population is going to be female biased. So it's kind of a weak link, but it's suggestive that that might be an area to study more. So here's the full distribution on planet Earth to date. It's not very many species that we know have Wolbachia, and these are not very strong records. We have very little data for those. So obligate mutualism, we know that Wolbachia occurs in the PPNs. The gene repertoire is more like that of mutualists, but we're not really sure. Perhaps it is an intermediate. Reproductive parasitism, well, we know that there's no riboflavin likely produced, but iron or heme synthesis may be part of its mode of operation. And so last, and I hope we're definitely getting down to time, so forgive me if I rush, we're going to talk a little bit about Cardinium, which is the second endosymbiont unrelated to Wolbachia. It occurs at the same time in the same nematodes. It's also widely looked at in spiders and mites where it has the same phenotype, cytoplasmic incompatibility, through different origins. No obvious shared evolutionary histories. So do we see CI genes, and what is Cardinium's role, and what's its distribution? And so I just added this image last minute because I thought it was pretty nice. This shows Cardinium cells from a paper in 1979 by Endo, where we have heteroautoglycines, and we have this now described as Cardinium buried within the rough endoplasmic reticulum of the esophageal glands. So it may be that this Cardinium has some role in the esophageal gland. So that's a question to be looked at. And here's our fish images. In yellow, you see our Cardinium cells. We see them also in the ovaries, and here we see them densely throughout the body. Just another close-up of that. We see them clustered. These are the developing oocytes, and we see Cardinium within those cells clustered around the nuclei. And this is just some genome pictures. So our Cardinium has a genome size that's somewhat small, but not as small as the only two other sequenced genomes from Cardinium, which are in insects. And so I'll skip over this gene plot and go to the phylogenies. Where does this Cardinium from Protolycus penetrans fall? Well, it's at the base of the tree from the full genomes, or kind of nested between a group from biting midges. But there's this, all in green here, we have this plant sap diet. So maybe there's a connection again with Cardinium and Wolbachia with the plant sap diet. When we do ortholog comparisons, there's a lot of information I won't go over today, but we see many horizontally transferred gene candidates in our Cardinium. So there may be lots and lots of genes that may be coming from outside of the host. I'll skip over this for the time today as well, so we can finish up. And we have metabolic capacity is mostly empty squares, so there's very little metabolic capacity. But let's go to, these are some horizontally transferred genes. Let's go instead to the distribution. This is the whole world, as far as we know it, of Cardinium. And not very many plant parasitic nematodes in that have been found to have cardinium. So I would argue that there may be more, particularly because our phylogeny showed, I skipped over that, but a long branch between our two that have 16S genes. This little bit of data here, I just also put this in today, is from the heteroglycine samples from some of the collaborators here, where we look at the coverage of cardinium. This is the number of copies in a given sample compared to the coverage of the host. So this is the host, this is the cardinium, and we see some decent numbers of cardinium. So heteroglycines indeed does have cardinium. We have PCR prevalence data, so we show the co-infection occurs. All the individuals with Wolbachia also have cardinium in our Protolingus populations, and we have some numbers that won't go into now for time as well. So these are images of those co-infections. We see in yellow cardinium and red Wolbachia, and we see them occurring in similar parts of the nematode. Similar images, and we see the esophageal glands here. Are in this section here where we don't see cardinium, we see some Wolbachia around the outside. When cardinium's by itself, it does seem to occur in the esophageal glands. This is a model of potential cooperation. It's very tentative right now. So there's some complementary missing genes in these two endosymbionts that co-occur. I'm going to skip over that and the experiments in the lab. We can talk about that later. So we'll just finish up. We do see that symbiont genomic repertoires can allow a host to explode new plants. We don't know the answer to this, whether loss of symbionts drives alternate plant use or alternate plant use drives loss of symbionts. Horizontally transferred genes from the microbes can affect plant use, and that's been looked at extensively in many systems. Also, past horizontally transferred genes may facilitate future symbionts. And so I am looking for graduate students. If anyone knows, this is a building out my window that's being built to house all of the grad students who want to come in and work with me. And here's my students who've helped me. And these are the teams that they fall into, a PPN team, insect team, and I work on a lot of other things. So thank you very much for your time.
Questions for Dr. Brown?
So, like with these phloem-feeding insects, so the phloem has very low amino acids. So then they rely on the symbiont. But where does the nitrogen come from?
I was just talking to one of you today about this. I don't have that answer really. We were explaining there is enough nitrogen apparently, but I wonder if there's something missing that we don't know. That's very mysterious. But I know. Did you see if there was a recent paper where that was looked at?
I think Angela Douglas has something.
Angela Douglas. So we'll have to go see Angela Douglas's work for that.
From the phloem.
It's in the phloem.
Yeah,
There's
no,
got to be
acids in the phloem.
But who's making it and what form is it in? I guess what we're looking for, right?
The bacteria have a way of converting that into amino acids.
Yeah, they do. They do. So they can use
whatever
they're de novo synthesizing amino acids. So they have limited options on them.
Further questions?
So the leaf hopper that you looked in cotton, right?
Mm-hmm.
Do you look at the same species? That one was one of these.
Yeah,
it
again,
has very
yeah.
generally. So it's the same in different hosts.
So this is a species complex again. It's the Empoasca genus is kind of a bit of a mess taxonomically. There's hundreds of species in the genus that look kind of hard to tell apart. So our current strategy is just grab all of them, sequence a bit of the insect to give kind of a marker of the insect genotype, and place that with its feeding type if we can gather that information and its symbiont status.
Just some questions. Oh, thank you.